session is Tyler Bourret, formerly from the Rizzo Lab at UC Davis, and now at Washington State University, Puyallup. And this is what I like to call the, oh no, not more of them kind of talk. But if there are more of them, and it appears there are, I have infinite faith in Tyler to identify them.
Thanks. And likewise, you've already heard from Ted about this Santa Clara Valley Water District survey that you're going to see furnished a lot of these novel species. And in fact, we've just kind of wrapped it up, or maybe midstream in the last SOD symposium, and I may talk about it. I'm still sorting through some of those species. I want to start off with, this is the SOD symposium. So much of this huge change and abundance, new abundance of Phytophthora species, I really think was spurred by sudden oak death for a bunch of reasons, so credit goes to a lot of people in this room. Before I dive into all of these species, I kind of want to talk a little bit from the systematic perspective, which is not anyways, that's my take on things. It's not particularly fancy, but so why do we have all these provisional species? Well, there's actually a good reason for it. It's not just laziness on the part of people like myself. We are in an era where if you study eukaryotic organisms, plants, fungi, fungal-like protists, we believe that species are real things. We can go out and measure them. And we still don't really know, I would say, how to measure a Phytophthora species because we don't really have a sense of what a Phytophthora species looks like in its native habitat, what its population genetics really, what we expect them to be. So we're in a really difficult situation where we're finding all this novelty, but myself as a good scientist, I can't publish something that I'm not sure might be a real thing. So that's where we have these provisional taxa. This is Phytophthora. We can't just sit on this. We need to talk about this. And we've already heard about why it's important to communicate about these things, whether it's on a species level, a subspecific level, a DNA level. And so we essentially have a de facto phylogenetic species concept. If you have a novel ITS sequence and COX-1 sequence, which are our two barcoding loci, then that's good enough evidence that you should at least get this information out, get the sequences into GenBank, let the world know about this, and let somebody else come in later and lump all these species together. That's famously what Grace Waterhouse did back in the 20th century, British mycologist, the early period of phytophthorology, where everyone was like, every new Phytophthora coming off of a new host is a new species. And no one was growing them and looking at them, and she came in and lumped them all together. So someone can do that later, but in the meantime, we need to talk about these things. So that's the reason, in my opinion. There are other reasons why we have provisional taxa, but that is my main reason why they're legitimate. They're not something to ignore. So, getting into my new species, before I get into Phytophthora, I want to talk about Nothophytophthora. And I'm going to go back to the last SOD symposium. So I had a poster summarizing the results from the Rizzo Lab stream monitoring project, which started in 2004, still ongoing. As we were sorting through some isolates, we found what after a few different gene sequence, was like, this has got to be a new genus. And it comes between Phytophthora and Halophytophthora, which is Phytophthora's closest relative, at least in sort of this direction. And so I put a little blurb just in the corner of my poster 3 years ago, and I said, this is a new genus, and it's Hydrophilomyces for water leaf fungus, because we got it from stream dating. So fast forward to a few years later, there's been a lot of work in these genera that come between Pythium and Phytophthora. And so Halophytophthora is one of the older ones. This great, one species of Halophytophthora got raised to a genus, the chalice bearer, which I just think is such a fabulous name for an organism. And then Nothophytophthora got recently described as well. And what's interesting is really we have not associated any of these species or genera with any really big landscape disease or agricultural disease. I'm not saying they're not pathogenic, but it's worth noting. So, just a big picture I won't spend too much time, but just a big picture of the Paradox borealis. We've got Pythium down here. The gray boxes are what we would call Pythium, sort of the old school Pythium, or if you're just looking at something on a plate, this is what we would call Pythium. But we've got Phytophthora, Nothophytophthora, Phytophthora, and then of course, there's 20 genera of downy mildews that are really just specialized Phytophthora species that are all mixed in. So, what about Hydrophyllomyces? Well, it turns out Hydrophyllomyces was Nothophytophthora. And I didn't do anything with it. I was working on dealing with all these other new species and getting my PhD finished. So, the first species, the one we got from rhododendron leaves and stream monitoring, that is Phytophthora, I guess I left it out of this phylogeny, Nothophytophthora caduca. And this other species that Kamyar Aram got from a bay leaf bait that you put into a river is definitely a novel species, not one of the 6 species. I can't believe I left off caduca here, but it's not one of the original Nothophytophthora species. Okay, getting into Phytophthora. This is the only one that I've actually published anything about that we found. This is from the big Santa Clara Valley Water Survey. Apologize for this, you're not going to be able to read this, but this Phytophthora taxon mugwort, this is like a pure new species. So when we got that first barcoding sequence, it's nice and clean, you blast it, which is when you compare it against all the sequences in GenBank, 93% match. And it's a great moment where you know, I just found something really new. And it was really new, so new that we had to create a new clade, subgeneric clade to accommodate it, number 13. This is the part where I had a whole slide about what are subgeneric clades that I'm not going to subject you guys to. So we also found another one that seemed to be a new clade, Phytophthora taxon juncus. What was so cool is in this SCV-WD survey with Phytosphere and Rizolab, we sampled some juncus, some rushes. That was the hottest genus. That was the genus with the highest potential of Phytophthora. And people don't often think of monocots as Phytophthora hosts. So we had this cool species. We had just baited it from the soil underneath the juncus. We didn't know anything about it. Wait a couple years, and somebody described Phytophthora lilii. This is from Japan. Phytophthora megasperma is this grab bag genus where like half of the genus of Phytophthora, if you key it out morphologically, you get megasperma. So the Japanese were sorting through all these things they had deposited called megasperma, and there was all these ones that are associated with Easter lilies. When they did pathogenicity tests, they seemed very specific to lilioid monocots. And so we have a potentially new wing and potential monocot inhabiting or infecting group of Phytophthora. It was cool. I got to meet one of the people who worked on this last summer in Boston. Just while it's up here on the screen, another one that we found from that same survey, and this may be the same as the Aph Europea that Ted mentioned, is Phytophthora sp. cadmea. It's in this Europea species complex. Oh, we found a hybrid species in Guadalupe River. Same survey. If you want to know more about how we detect hybrids, look at one of my posters later tonight. From the stream monitoring results that the Rizzo lab had, found a new species in the Citricola species complex. And I tell you, you probably will not find another phylogeny that has all of the species in the Citricola species complex in it. But what's really cool is we baited this a couple times from the very southern end of our stream monitoring range, close to Rich in San Luis Obispo, but it was also baited by OSU, and this is a new species in this complex. And I just want to point out, it kind of looks like this very difficult species complex is all nicely sorted out here. But if you look at this, that's a multilocus phylogeny from beta tubulin, COX1, and ITS. Really, only the COX is got any sort of decent separation. You can separate the species out with those other loci, but they're very close. This is essentially one base pair difference, this little thing there. So they're really closely related. COX1, it's a powerful gene. What COX1 is in GenBank, there are 3 different loci people call COX1. They only overlap at about 150 base pairs. It's a huge problem. I tried to bring it up in this big OMIC meeting last summer, got shut down, so it's going to continue to be a problem. So besides surveys that I was doing myself, I got into a great position where the Rizzo lab was receiving a lot of isolates, either from CDFA projects themselves, like this one with taxon wisteria, new associated with wisteria, which is a legume. What's kind of cool is this is Phytophthora soji, probably the most economically important Phytophthora in the country, and Pisai and Asiatica. Asiatica is associated with kudzu, which is another. So this is a leguminous wing of Phytophthora. Okay, that totally set up to talk about taxon agrifolia. So I won't even talk that much about it because Ted talked about it. We've been working on the species. I got an isolate early on. I was working on the phylogenetics. It's definitely distinct. Then last year, in some Angeles National Forest surveys, we found another related species. So these are a new subclade of clade 8, taxon agrifolia, and I apologize for calling it taxon agrifolia too. They're very distinct. Clade 8's a really interesting clade. We've got some things we're very worried about, very common restoration species, so one to look out for. And as Janelle mentioned, I just moved labs, and on my first week in the Chastagner lab, I was asking one of the technicians, you got any weird, problematic Phytophthora sequences? She said, absolutely. And here it is from lavender, this is agrifolia 2. So another state, another associated host. So with whatever time I have left, this is my last species or paraspecies, but this is one I'm going to make the case for why we need hardcore nerdy systematists working on Phytophthora, even though it's just not a very sexy wing of biology. So Phytophthora cultans and terminales were described in 2015. They're in what I would call the Citrophthora species complex. Based on phylogenetics, and also cultans was Citrophthора's closest relative, and they also distinguish it based on sexual characteristics. So here's the phylogeny of cultans, apologies, and Citrophthora. So how about in California? Well, we found Phytophthora cultans in the same survey I keep talking about. When we've been doing these surveys in Angeles National Forest, or also looking at isolates from other people doing those surveys, found another species in the Citrophthora complex. And I couldn't say it was Citrophthora, but it wasn't the same as the cultans. And I was kind of unclear. I gave it this taxon erudiction. Thanks to APHIS, now we can sort this out. So Gloria Abad and a lot of other people from APHIS, including John Benapple, who is a Southern Oak Deather and a Risonian, they really had a monumental effort that just sort of culminated in 2018 to get all of these sequences from the X-type strain. So the official version of that particular species, if we need to divide up the species, which we're certainly doing, or if we're not sure, if we key out some Phytophthora species and things that we keyed out as the same thing are phylogenetically distinct, where does that species go with? So that's why we need the X-type strains. Doesn't solve all the problems, you still have to deal with species boundaries, but it can really help. So what did we find? Well, it turns out the X-type strain of Phytophthora citrophthora, which was not included in this original study, it comes out right here. Phytophthora citrophthora X-type and a cultans X-type are indistinguishable based on every gene that have been sequenced. In other words, Phytophthora cultans is an invalid species. Just stop using that. So how the heck did this happen? So this Dutch group, led by the researcher Manon Veld, when they were distinguishing the cultans from its closest relative Citrophthora, they didn't actually go out and get the official version of Citrophthora. They didn't even get things that might be closely related to it. Citrophthora is described from California more than 100 years ago. We've already heard about it from Steve's talk, from the citrus etching. It's the same group of people. And so it was at least described from the New World. So here's the original description. It was originally described as Pythia cystis back in the early days, abundant in winter and spring in Southern California lemon orchards and packing houses, Casasiris blossoms. Then this great American phytophthorologist, Leon Leonian, came through a couple decades later and officially typified it and deposited a strain. So that's the one that's the same as a Coltens. I love this thing. This organism is so obviously a Phytophthora species that the genus Pythiocystis is gone. So, what about the morphology? They said, well, it's not just, it's phylogenetically distinct and it's morphologically distinct based on sexual characteristics. You know, our two species are different, and because Citrophthora is sterile. If you go back to the original description of Citrophthora, this is from, I believe, the 1905 people. This was kind of before oospores were really a thing. And all of our descriptions of Phytophthora species and the different genera were all based on sporangial morphology. So nobody in the original description mentions anything about oogonia or oospores. They're not drawn here. It's not because they couldn't find them, it's just wasn't part of Phytophthora taxonomy at the time. So this idea that, oh, our species is different because ours is homophallic and the other one's sterile, it's just based on lack of sort of precision, I will say. What about this one that they were calling Citrophthora? Well, that's taxon Areodictyon. So both of these two species, there are two distinct species, they're both associated with restoration in California, and now we can at least, you know, try to talk about them in some correct terms. And that's it. Thanks for everybody. This group of sort of collaborators is definitely a big part of why I got to be at the center of all these fun new species of Phytophthora.